Hi, welcome back. Chapter 3, Biological Macromolecules. Last time we left off with the importance of water and some of the chemical qualities that make water so unique. The extensive hydrogen bonding is what makes water a cohesive molecule, which means it likes to stick together. In the water molecule, the slightly negative oxygen attracts the slightly positive hydrogen. Now we can apply the knowledge of how molecules are formed between the elements and study the very foundations of biology and take a look at the structures of sugars, fats, proteins, and nucleic acids. So chapter 3 shows us the key molecules that form the foundation of all organic life on Earth. All living things are organic because they contain the element carbon. We use the term organic a little different when learning biology. Remember in Chapter 2, we looked at how the sharing of electrons in an outer orbital can form one of the strongest bonds in nature? Do you remember what that type of bond is called? Well, carbon tends to form those bonds quite readily. In fact, when we look at the bonding patterns of carbon, we can start to see why carbon is the backbone for all organic structures, and today we are going to focus on the four major types of macromolecules. Before we dive into our structures of sugars, fats, proteins, and nucleic acids, you may need to familiarize yourself with some of the common scientific notations found on this page in your notes. Common examples are listed for you here at the bottom. These include the notation for the hydroxyl group we discussed during the last lecture in the context of pH. The OH groups in water create an alkaline solution. As we get into the organic molecules like proteins, the amino group has its own notation, NH2. And another common one is the phosphate group, PO4. These are highlighted for you in red. Polarity in carbon-based molecules is important only when you consider that water is a polar molecule and only water-soluble substances can mix with water. When we try to mix a polar solvent with a non-polar solute, they don't like each other and they don't mix, just like oil and water won't mix. They have different polarities. Non-polar carbon-based structures often take on a ring shape, and there are a few different ways to draw these out. Organic molecules can form long linear structures like this one shown here. This is what the chemical structure fat looks like. All the carbons link together forming a backbone, and they are fully hydrogenated. This is called a saturated fat. Linear fats are also packed together tightly at room temperature and form solid fats, and they look often like white, shiny, maybe something that you've seen in the bottom of a frying pan after cooking meat. So let's get started on our four key macromolecules. Macro because they're large. We already saw how large fats look with their long chain of carbons linked together in series. Here in Chapter 3, you're going to learn about carbohydrates, which are simple sugars containing C, H, and O for carbon, hydrogen, and oxygen. These are used as quick energy by organisms. Lipids or fats are also referred to as triglycerides. They're also made up of C, H, and O, but in different ratios than sugars. And we already saw that the carbon-carbon series that forms the backbones of all fats in the previous diagram. Proteins are made up of the 20 amino acids that act as the building blocks for functional proteins. This is a very diverse group, and protein function is based on its 3D structure. And we'll take a look at those configurations here in just a minute. And the fourth and final group is the all-important nucleic acids. This group of molecules are all made up of a sugar, a phosphate group, and a nucleotide base. The main function of a nucleic acid is to transfer energy or store the entire genetic code of the organism. You may already be familiar with the structures DNA, RNA, and ATP. These are great examples of nucleic acids. These four macromolecules participate in different biological reactions that sustain life. These are highlighted for you in red. Most of what we eat will provide these important molecules to us. Organisms that consume food can break down these molecules as well as assemble them into larger groups. Large macromolecules with their complex structures can undergo hydrolysis reactions. Hydrolysis reactions is how large polymers are broken down into smaller monomers, and in this case, a dehydration reaction. The small single monomers can become linked. When we use the term metabolism, we're referring to the sum total of all the chemical reactions that take place in an organism. We can visualize these reactions by first taking a look at the hydrolysis reaction. This term literally means to split with water. We see how we can break the bonds between the purple and the blue molecules here on the left. To the right, we've added a water, and the two molecules are now separated. Your digestive system breaks down macromolecules, food, into monomers, like amino acids, the building blocks of proteins. The cells then take these monomers to construct new polymers. Polymers are also known as tissues, like muscle. The dehydration synthesis reaction, also known as a condensation reaction, is where a hydroxyl group, remember that's the OH, is removed from one monomer and a hydrogen from another, producing water as a byproduct. In this case, we are joining two smaller monomers together to form something bigger. These reactions will form large, highly branched, diverse chains that can serve as the backbone for a wide variety of other important compounds that we will look at throughout this semester. Carbohydrate sugars are usually 5 to 6 carbon molecules that can form simple to complex structures as they become linked together, and some form branch structures. These sugars shown here are examples of simple monosaccharides. Glucose, fructose, which is the natural sugar found in fruit, galactose, deoxyribose, and ribose, those are all simple sugars. When a simple monosaccharide links up together with another monosaccharide, there is a glycosidic bond formed between the first and sixth carbon on the two molecules, and they share an oxygen between them. This is a covalent bond and is therefore a very strong one. When these bonds are broken, it is an energy source for the consumer. Complex carbohydrates tend to look complex and may have extensive branching. Examples include glycogen, which is the stored form of energy in your liver, starch, which is produced by plants for energy storage, cellulose is another one, which is often referred to as dietary fiber, and chitin, which is found in the exoskeleton of insects and other arthropods. Chitin is also unique because it is the only one of these complex carbohydrates that contains a nitrogen group. Now let's go back and learn a few more things about fats. The proportion of oxygen in fat is less than what's found in carbohydrates. All lipids are hydrophobic or water-fearing, so they do not dissolve well in water. Fatty acids can be either saturated, which lacks any double covalent bonds, or unsaturated, which means the lipid structure will have one or more double bonds. These are sometimes called pi bonds. This is highlighted by the red box on these carbons that have two lines drawn between them. So fully saturated on the top, and the lower structure is an unsaturated fatty acid. There's a pi bond between the two carbons. Biologists need to consider these four main groups of fatty acids. All of these are hydrophobic, so they won't dissolve well in water. These characteristic long carbon-carbon chains are what make them immiscible in water. All triglycerides have a glycerol head group, shown here on the left and highlighted in brown, with 3 fatty acid chains attached to this group. Thus the name tri, meaning 3, glyceride. Phospholipids are similar in structure, but we'll see that one of the fatty acid chains gets substituted for a phosphate group. This is a very important molecule when we consider the cell membrane. Eicosanoids, I'll just mention them here, but we won't use them very much in this course. They're needed in cellular communication, specifically the inflammatory response during injury or allergies. Steroids. These are hormone structures and will take on a four-member ring-like structure. These molecules are very powerful and are prominent methods of cell-to-cell communication in mammals. We'll talk about these again in Chapter 5. Here's the reason why we covered a bit of chemistry in Chapter 2. Reading this diagram from left to right, we have a carboxyl group in blue and is always found at the reactive end of a fatty acid. Then we see the characteristic long chain of carbon-carbon-carbon bonds. And this is a fully saturated fatty acid, or is it unsaturated? You can email me your answer. Remember, you can pause this video at any time. And at the far end, opposite to our carboxylic end, we have a CH3 group, and this is called a methyl group. Let's take a closer look at our phospholipids that we just mentioned. Because the phospholipid is so important in biology, let's take a look at it a little more closely. We will study the structure and its important function in the next chapter when we study cells. The three colored regions highlight how there's a substitution for a phosphate group here in blue. So that means there are only two fatty acid chains instead of three when we looked at the triglycerides earlier in this lecture. This blue phosphate group allows part of this molecule to be water-loving or hydrophilic. So the take-home message here is that this unique biological molecule has dual solubility. And we're going to talk a lot more about this guy coming up soon. Now let's look at how this molecule performs in water. Notice that the fatty nature of this molecule makes it take on either a bubble formation called a micelle here on the right, or a layered structure similar to what you'd see during an oil spill in the ocean. Now this bilayer allows for the fatty acid chains to hide from water, which is on both sides of this layer. Fatty acids are hydrophobic, but the phosphate groups are fully soluble in water and happy to be in direct contact. These structures form functional aspects of the cell, either as a barrier in the bilayer type of format or the transport in the bubble micelle configuration. We'll look at both as we move through the semester. The term we use to describe dual solubility is amphipathic. That's one of your red vocab terms here on this slide. Now, for our most diverse group of macromolecules, the amino acids, there are 20 different amino acids, and they always work together by linking up and forming big functional proteins. We will talk about how the body makes proteins more in the next units, but amino acids can be small or rather bulky, but they always have an amino group here on the left, highlighted in purple, and there's a carboxyl group on the right. Remember where you saw that before? And then the R group in green is what changes every time we talk about a different amino acid. For comparison, and you can get yourself familiar with all 20 of these amino acids if you go ahead and refer to figure 3.23 in your ebook. So proteins are only functional if they are made properly. So we have primary structure, secondary structure, tertiary structure, quaternary structure. It just depends on how much folding is happening. So at the top here, we can see how several amino acids are linked together through peptide bonds, and they look like a ball and stick model. The same folding has to happen in order for them to be functional. There's sort of this Shirley Temple curl down here in the lower left. That's called an alpha helix. We see this helical shape in DNA also. And more folding can form a beta pleated sheet, and we can get even more folding to get a globular structure like what we see in hemoglobin. You have examples of these in your full set of lecture notes. One of the most important functional classes of proteins that we will run into a lot in this course are enzymes. Enzymes work to push chemical reactions forward towards product at a faster rate than without these specialized proteins. This is one of the keys to regulating our metabolism. There is high efficiency and energy is not wasted on products the body does not need. Enzymes are highly specific for running usually only one type of chemical reaction, and when we take a look at proper enzyme function, it will remind you of how a lock and key work together. That analogy is used because enzymes always work with a substrate. Substrates are the limiting reagent in an enzymatic reaction. The enzyme will bind specifically with its substrate in order to make the product. The enzyme E, up here at the top at 12 o'clock, can only bind with substrate S. E plus S, that means the enzyme has now combined with its substrate, form a complex, and notice here, down here at 6 o'clock at the bottom of the circular diagram, the substrate gets used up in this reaction. S gets converted to the product P. S is the limiting reagent. When it runs out, the reaction stops. So there must be an abundance of S in order to keep making product. We will practice this in one of our future lab experiments. Okay, our final category, nucleic acids. These are DNA, RNA, and ATP. These are also very important macromolecules in biology. The double helical shape of DNA here in the upper left carries the genetic blueprints for every organism. RNA is a close cousin to DNA. In fact, RNA is a faithful copy of DNA. Notice it has a single strand of genetic material. Its job is a little different because RNA actually gets to leave the nucleus and it goes out into the cytosol of the cell and finds the machinery involved in protein synthesis. In your ebook, figure 3.31 illustrates the similar structures between these three forms of nucleic acids. Notice they all have a 5-carbon sugar, a phosphate group, and one of 5 different bases shown here on the left in green: cytosine, thymine, uracil, adenine, and guanine. ATP stands for adenosine triphosphate, and it's unique because it has a triple phosphate extending off its structure. But that's because ATP provides energy and is not responsible for carrying any genetic code. ATP's universal energy carrying capacity is due to its unique triple phosphate bonds highlighted here on the left in purple. These are high energy bonds, and when they're broken, that stored energy is released for cellular work like locomotion, transport of molecules, and chemical rearrangements. Cells can't use that cheeseburger you just ate. Cells can only use ATP energy. We will have a chance to talk a lot more about cell metabolism in Unit 2. Please rely on the assigned reading pages in your ebook according to your syllabus lecture schedule to identify these important macromolecules that we will be using in great detail throughout this course. And also, in addition to reading your book, use the full set of lecture notes that have been provided to you. Okay, see you next time.